In this video, we will build an intuitive picture of how a non-competitive inhibitor decreases the rate of an enzyme reaction. Here is the question. How does a non-competitive inhibitor decrease the rate of an enzyme reaction? Is it by binding at the active site of the enzyme, by changing the structure of the enzyme, by changing the free energy change of the reaction, by acting as a coenzyme for the reaction, or by decreasing the activation energy of the reaction? We will eliminate the wrong options visually, then see why the correct choice is by changing the structure of the enzyme through an allosteric binding that lowers Vmax while Km stays the same. First, non-competitive inhibitors do not bind at the active site. They bind at a separate allosteric site. Next, they do not change the free energy difference, delta G, between reactants and products. They are not coenzymes. Coenzymes are helpers that participate in catalysis and are often organic molecules. And they do not lower the activation energy the way a catalyst does to speed up a reaction. They actually reduce the catalytic rate. The best description here is that they change the structure of the enzyme through allosteric binding. Let us build the visual intuition. Here is an enzyme shown as a green blob with a yellow active site pocket and a blue substrate that fits that pocket. The substrate binds the active site and is converted into product quickly. Now, a non-competitive inhibitor binds to a separate allosteric site, shown in purple, not the active site. This binding changes the shape of the enzyme, including the catalytic geometry, so the enzyme works more slowly. Importantly, substrate binding affinity, Km, is not reduced in the pure case. The substrate can still bind, but the catalytic step is less efficient, so Vmax is lower. On a Michaelis-Menten plot of rate versus substrate concentration, non-competitive inhibition lowers Vmax but leaves Km unchanged. We will draw the control curve in green and the inhibited curve in orange with a lower ceiling. The new Vmax is lower. The Km value is the same, so the vertical line at Km hits half of each curve's own Vmax. That is why the apparent affinity stays the same, but the maximal rate decreases. The key outcomes are Vmax goes down, Km stays the same. The mechanism is allosteric binding that changes the enzyme's conformation. That structural change reduces catalytic efficiency of the enzyme-substrate complex without preventing binding. Therefore, the correct choice is by changing the structure of the enzyme.